Hey class, so today I'm going to review with you the basics of enzymes, and this is on page 66 through 69 in your IB biology book. So enzymes. Enzymes are proteins, and they are protein catalysts. And a catalyst, and I hope you learned this from last year, a catalyst is a molecule that speeds up a chemical reaction. The reaction would happen by itself, but it may take a really long time. So a catalyst puts the molecules that are reacting in an appropriate position so that they will react more quickly. So on page 66 and 67, they refer to two analogies, and one is the glove and the hand. The glove is the enzyme, and the hand is the substrate that binds to the active site in the enzyme. So there's a few vocabulary words that we need to review. So an enzyme is this protein that does the work, that helps put everything in the correct position so that the reaction can happen. And there's a picture on that page, on page 66, which shows something like this, right? Except it looks a little bit more chemical-like. And this is termed, right there, that is termed the active site. That's where the action happens. That's where the substrate binds. The substrate fits perfectly into the active site of the enzyme, so some kind of chemical reaction can happen. This is called the substrate. And as you know, your hand fits inside the glove perfectly, and then, in theory, a chemical reaction can happen. Not in this analogy, but you get what I'm saying. The other analogy that often is talked about how enzymes work is the lock and key model. So the lock is the enzyme, and it fits a specific key, right? Only one kind of key fits in a specific kind of lock. So that would be the substrate that's acting upon. So both of these models are important in understanding how substrates and enzymes fit together. And remember, a catalyst speeds up a chemical reaction, but it doesn't get used up in the reaction itself. So an enzyme can be used many times to do the same process. So in the next section here, they talk about the effect of temperature. So in this graph, which I recreated from the one in the book, as you increase the temperature, as you go right on this graph to the right, the rate of the reaction increases because, as you know, when you increase temperature, the molecules move faster. So when they move faster, there's a greater chance that you're going to have them collide in the appropriate way. And that reaches a maximum of some kind. And then after that, the protein starts denaturing, meaning unfolding. And when the protein starts unfolding, it can't do the action. So think about in the previous picture and on the previous slide, where if this protein isn't folded correctly and the active site isn't just right, that substrate won't fit in there. And that happens when you increase the temperature too much. It causes denaturation or unfolding of the protein. In the next section after that, on page 67 and 68, it talks about the effect of pH. Now, when you draw your enzyme, right, my little crude little enzyme, there are certain charges that'll be along certain amino acids that are along the active site, and that has to match perfectly the substrate which binds into it. If you change the charge, for example, if you change the negatives to positives, right, and there's positives along here on the outside of the substrate, and initially there's negatives, right, so it fits perfectly, right, they attract together. But if you change the pH so that the charge becomes positive, now the substrate will not fit in the enzyme's active site. So changes in pH can affect the activity of your enzyme. Now this graph shows that as you increase the pH, you can affect different enzymes in different ways. So this enzyme works at a low pH. It has its maximum rate at low pH. So that could be like an enzyme in the stomach, right? Because in the stomach, the pH is very low. So as you decrease the pH too much, or increase the pH from its optimal pH, optimal means maximum, you get drop-offs in the activity. But most enzymes work around neutral pH, which is around 7 here. So if you increase the pH towards basic, the activity drops, and if you decrease the pH towards acidic, the activity drops. So this could be an enzyme that could be anywhere where the pH is a neutral pH in your body. In the last section, they talk about the effect of substrate concentration. Now these brackets, what they mean is concentration. So instead of writing the word concentration down, I just put the brackets around it, and hopefully you learned that in chemistry last year. So in this graph, they show as the substrate concentration increases to the right, the rate of the reaction increases until you reach a maximum rate. So in the toothpick ace lab, you're only able to break a certain number of toothpicks, and after you add too many toothpicks, you can't break them any faster. So if you increase the amount of substrate, right, you increase more of them, and they fit perfectly into the active site, except that the enzyme can only work so fast. So in this area of the graph, it hasn't reached that maximum rate yet. But at this level, here, we've reached the maximum rate, and the enzyme can't break down the substrate fast enough. In the last section of that's on page 69, it talks about how, well, a lot of people in the world, right, everybody, starts off being able to break down the protein lactose, right? It's a disaccharide, as you memorized earlier. And as an infant, you could break down your mother's breast milk. But as you get older, the enzyme that breaks down the lactose, it's called lactase, doesn't get made as much. And then when you drink milk, the bacteria inside your stomach are actually breaking down the milk instead. And that causes things like gas and diarrhea. Lactase, sorry. So what you do is you can pre-treat your milk with lactase, right? So we're going to do pre-treatment right before you drink the milk. And it breaks down the lactase into its monosaccharides. And when you break down the lactose into its monosaccharides, you can now be able to digest it because your body can digest the monosaccharides but not the disaccharide. So make sure you please write some questions down that this video brought up in your mind, and we will address them in class on Wednesday.